Okay, let's get started. So it's an honor to introduce Bruce Walsh. When I was in grad school, sort of Walsh was one of these names that you saw a lot in main papers and so many books as well. And now he's writing how many volumes, Bruce, for the new book that you're writing?
Just two more.
Just two more, right? Two more volumes that are coming up, and he's been working on them for years. But Bruce is one of the foremost authorities on quantitative genetics in the world at this moment, and it's really fun to have him here and teach us many different things, starting from the basics all the way to where things are going now and where new advances are being made in quantitative genetics and understanding how to analyze new data that's coming up um, in light of older methods as well. So without further ado, Bruce.
Thank you, Deepa. So first of all, it's been really fun to be here. It's been exciting to work with the exceptional students that are here. In spite of the fact they tend to ask so many questions, we can only cover about 3/4 of the material. We'd much rather have that than people who just sit there and do anything. So what I want to do basically is give kind of a overview, kind of a history of science perspective about the search for quantitative genetics. We're going to go all the way back to Mendel and see how some of the connections are tied together. And so this is going to be kind of a non-technical, just sort of overview that takes us from sort of the roots of quantitative genetics up to sort of the current controversies. At least that's the plan. So basically, if you look around the room, you see individuals differ in trait values, right? Pick any trait you want, there's variation. And so the question is, well, obviously some of that variation is genetic because it runs in families. Some of that is environmental because individuals with the same environment often look similar. How do we decouple that? And more importantly, how do we find the genes underlying that variation? And what's their nature, right? In particular, how much of the phenotypic, that is trait variance, we see as genetic? How much of that is usable? That is, as we talked about genetic variance, only part of that is passed on from parent to offspring. And that has implications for evolution, breeding, and medicine. Is that genetic variation due to a few major genes or a large number of so-called polygenes, a small effect? What's the nature of that variation? Is it additive? Is it dominance? That means an interaction between alleles at the same locus. Is it epistatic? That means interactions between alleles at different loci, nonlinear interactions. Are changes when you get mutations that give you variation in the trait, do those occur in protein or RNA genes that have function? That is, are they structural or are they regulatory? Are they in regions that control genes? And finally, just one of many questions, I think the pointer is fading fast here, is the between population that is fixed variation different from the population we say segregating within a population? So this paper appeared a couple of years ago. The classic paper is Fisher, 1918. And so in science, people are always trying to come up and lament that the field is dead, or the field has lost its way, or the field should be put down like a rabid animal. And they lament, in particular here, that quantitative genetics assumes the infinitesimal model. We'll talk about that, meaning that genes all have tiny effects. It can't account for the so-called missing heritability issue, that we'll talk about that as well, and other issues. For example, they incorrectly assume it only deals with something called additive variation, looking at alleles one at a time instead of combinations, the variance they account for. So what I want to address is, how real is this concern? And biology is unique in the sciences in that there's historicity involved. In the biological processes we see, they're shaped by by the tree of life, but also fields of science are shaped by their history. And ironically, much of this discussion here and these questions actually trace back to the very founding of quantitative genetics and the history of genetics in general. So there you go. You have to start with a picture of Darwin, and we'll get to a picture of Mendel in a second, and all our bases are covered. And we'll put Fisher in because if you're a statistician or a geneticist, that kind of covers it. So Darwin had a fatal flaw. His fatal flaw in his inheritance was his model was wrong. He assumed blending inheritance, kind of the model at the time. That, if you look at offspring, they kind of look like a mix of their two parents. And sex is messy with lots of fluids involved. So the thought of the time was that you basically take fluids from two individuals and kind of mix them, and you get kind of a blending as the offspring. So the idea was that the genetic material was somehow a fluid material. Now the irony, of course, is that Darwin in his library had an unread copy of Mendel. So if he just done his homework, he might have been able to figure this out. What's the problem with blending inheritance? Well, this is the first paper in population genetics. By the way, there's a little bit of overlap from Nick Barton talk for those that went to that. So Fleming Jenkins was a very famous engineer, Scottish engineer, and he had a little review, and basically putting his review in kind of modern terms, what he said was that if Z1 and Z2 denote the phenotypic values of the two parents, then the variance in their offspring is simply the average of those two under blending inheritance. If you assume these are from the same distribution and uncorrelated, that's just the variance over two. And that's the fatal flaw. The fatal flaw is with blending inheritance, you lose half the variation each generation. So without some mechanism to regenerate that variation, mutation, migration, whatever, genetic variation would quickly go away and there'd be nothing to act on. There'd be phenotypic variance, but none of those changes would be passed from parent to offspring, which is one of the critical assumptions for natural selection to work. So of course the answer was Mendel. And Mendel, key idea was genes were discrete particles. Since they're particles, if I have two fluids mixed together, I can't separate the two fluids. If I've got two particles, I can obviously pull them off and keep them intact. And that simple idea solves this problem of blending inheritance. So, but it's important to focus on a couple of other things first. So Mendel focused on a single gene that influenced a single trait. These were typically very discrete traits, tall or short, not subtle variation in between them, green or yellow, round or wrinkled, very clear-cut, very dichotomous traits. Sharp breaks occurred between phenotypes as opposed to kind of a continuous smear between. So in tall and short, there were no kind of intermediate plants. There was variation in short, variation in tall, but no obvious intermediates. Because you had particles, I could have an individual that had two particle copies, one of which overpowered the other in a sense, dominance. And that means there's hidden genetic variation. As those particles segregate, I recover variation, and it's this segregation variation that exactly solves this. So for example, under Mendelian inheritance, you get this, but you also get a term here that's actually equal to this, which is caused by segregation of the so-called heterozygotes that have two different particles. So under discrete particle inheritance, you don't lose that variation. But it took a long time for that concept to hook up, as we'll see with Darwin. So Mendel's work was not acknowledged for 40 years. Darwin was an instant success, it was all over YouTube. Mendel just kind of languished in newspapers until then. So in the interim 40 years between Mendel popping up and starting the acrimonious marriage with ideas of Darwin, there was kind of a whole independent field in genetics of continuous traits, which basically involved the foundations of modern statistics. And the classic paper is Francis Galton. Yes, he looks like this stodgy old Englishman, and he probably was. And he had a paper where he basically took the height of parents, averaged them, looked at the height of their offspring, and simply did a plot. That was the first regression. And so he was starting to measure statistical associations between one relative and another, and that's kind of the modern foundation of quantitative genetics. What he found, here's a graphical representation, here's his actual data replotted. So the black line is the best fitting curve. The one-to-one line is up here. So what you'll notice is that if you are an exceptional parent, up here, your offspring are, so basically this is the parental value, your offspring are less exceptional. Down here, your offspring are more exceptional. So if you're above the mean, your offspring tend to be closer to the mean. If you're below the mean, your offspring also tend to be close to the mean. He coined this the regression towards mediocrity, and this is where the term regression came from. And in kind of true British fashion, he basically said, this means that England will collapse into a singularity because all the smart people in England will have dumber and dumber and dumber children, and eventually they'll be all around drooling and nothing will happen. And in fact, of course, this is because they didn't know about segregation variation. So that's modern statistics, has its roots in trying to explain continuous trait variation. So unlike Mendel, he had a continuous distribution of trait values in the offspring rather than discrete classes. Mendel was rediscovered in 1900 by actually 4 or 5 independent groups almost simultaneously. At that time, There have been 40 years of people working on statistical methods of association, continuous traits. All of a sudden, this discrete particle school came up. As you might imagine, there's a bit of conflict between the two. And that conflict basically was driven more by personalities than by data. And basically, it took a long time for that conflict to sort of be resolved so we could actually merge the two of them. So let's go into that here. So basically, Weldon and Pearson, your famous Pearson product moment correlation coefficient, these were the supporters of Darwin. They felt that evolution was a result of numerous gradual continual genetic changes, and therefore there was a focus on small changes in continuous traits. So Mendel looked at short and tall peas. The biometrician said, well, there's this variation in height if you look in natural populations. There's no clear, sharp break in that, and that's continuous variation, and therefore evolution probably proceeds by a small effect series. So much of modern statistics, regression, correlation, ANOVA variances, trace back to the biometrician school. So we had the rediscovery of Mendel, 1900. You then had two groups almost appear simultaneously. The biometricians have been around a while. Evolution moves via very small steps. Statistical descriptions, continuous traits. Mendelians, discrete genes of major effects underlie evolutionary change, qualitative description, discrete traits. Vestiges of these two are here today. These are called quantitative geneticists. These are called molecular biologists. And we'll see how that connection comes in and the hopefully happy union between the two. So these people weren't dumb. They obviously must have had some evidence that made them think of one thing or the other. So one of the key bits of evidence that the Mendelians used to argue that small changes weren't important was work by Johansen in 1902. And Johansen basically took pure lines, we now know that means fully inbred lines, of beans, and he simply did a parent-offspring regression. So up here, that is, let's call it the diamond line. What he saw in the diamond line was no matter what the weight of the parental seeds, the offspring were the same. So unlike Galton, who saw a parent-offspring regression with a non-zero slope, when Weldon looked at that, within each line, that is this variation we saw within a line, none of that mapped onto the offspring. So within a line, there was no heritable variation, but between lines, there were heritable variations. So he interpreted this as major mutations and then noise inside that can't get transmitted. Now we now know these are genetically identical lines, so all the variation in here is environmental, and these are lines with different genotypes. So, but observations like this convinced the early Mendelians that continuous variation really wasn't that important for evolution because, according to this argument, it wasn't heritable. They never really went back and addressed what Galton was showing, but here, very clear evidence, very, very simple experiment that it wasn't heritable. Bateson is a central figure in this whole debate, along with Weldon. And we'll see in a second the importance of those two individuals, especially their epistatic interactions. So Bateson actually coined the term genetics. He was kind of the pit bull that said evolution was saltarial. It moved by giant jumps. It didn't creep along, it went boom, boom, boom. And their idea was these were the mutations that we saw occasionally rising. And that's, they said, where evolution largely occurred. He viewed much of, quote, normal phenotypic variation, the normal spread we see in traits. He viewed that as irrelevant to evolution. Ironically, he was a student of Weldon. Here's Weldon. Weldon was a biometrician. The antagonism between these two basically polluted any chance for reconciliation, right? We often think that, well, science is driven by, logical and rational men and women that sit down and take all the facts in front of them and struggle with their own views on something, and then finally are overwhelmed by the goodness of the data. In reality, as you know, science can be a bitter political thing. And my best quote in science was from Kissinger. And Kissinger said, the battle in academics are so vicious because the stakes are so small. And you often see that. And so this is a classic case where you basically had key ideas were sitting in isolated camps that only got together when one of the antagonists, Weldon, died. Not recommending that if you get into a scientific dispute, but just be aware that science has this element of politics that pervades it we often try to ignore, but but it's really there. So his death removed most of the personality issues that sort of blocked any attempts at the synthesis. And now the synthesis starts creeping together. Discrete genes, particles, discrete traits, statistical variation of continuous traits. Is there a happy middle ground? Key paper, Nilsson-Ehle, 1909. He was working with seed color in wheat. And he had a really bright red seed color and a pale seed color. We now know it's caused by 3 genes, each of which adds additional pigment. So he was able to basically show by crosses, you could get kind of a continuous distribution in crossing these, and yet kind of break down what it was. So he didn't get at the gene, but he basically showed you can get this variation. And so what basically his early work did, this was one of the first attempts that tried to bridge the major gene view of the Mendelians and the continuous variation view of the biometricians. The paper that it was this, Fisher, 1918. There's lots to say about this paper. Number one, don't read it. It's unreadable. How unreadable is it? In 1968, on the August 50th anniversary of Fisher's paper, Fisher went to Australia near the end of his life. The Australian Journal of Biological Sciences had a special reprinting of it, where they had each page was like this, a paragraph, 12, 14-point font from Fisher. The rest of the paragraph in 5-point font, just what the hell we think the above paragraph meant, right? Fisher is not a clear writer. Fisher could think in 4 dimensions, and his writing shows that, right? It kind of reflects that. But this paper here, by the way, this also warms my heart. He submitted it to the prestigious Royal Society of London. It was rejected. He had to settle for the Royal Society of Edinburgh instead. By the way, Edinburgh is regarded as the home of quantitative genetics. So they had a good start with this paper. But in this paper, as kind of to get to his result, so he did a couple of minor things. He showed how Mendelian genetics with discrete particles and continuous variation of the biometricians could be fused. The term variance first appears in this paper. Oh, and by the way, he developed the analysis of variance in the paper to get things done. So incredibly influential paper in both genetics and statistics, and rightly so. This is the founding paper in quantitative genetics, and it's a paper that starts to bridge discrete gene view, continuous variation view, and we need that to really understand how evolution works. So the importance of this paper really can't be overstated. It's also important to know what was Fisher's degree at the time. So, So in this paper, Fisher used a model, and this model kind of resonates today, rightly and wrongly, for several reasons. He used what's called the infinitesimal model. And Fisher assumed that your genetic value, so your phenotype was some genetic value plus some independent environmental noise. And your genetic value was composed of the contributions from a large number of genes, each of which individually had a small effect on the variation. Taking to the limit, that's a so-called infinitesimal model. You have an infinite number of genes, each of randomly small effects. He didn't assume that. Basically, that's kind of what the model leads to the limit. You have a lot of genes, all of whose effects are small. And this model served the base model for quantitative genetics for many decades. So the question is why? Well, it actually does that for a couple of reasons. Number one, we certainly know there are genes of major effect out there, but they're not super common. So if genes of major effect were important, I should see gaps in the distribution of phenotypes. We typically don't see that for most traits. Obviously, there are exceptions. I mean, obviously, Mendel found them, so they're out there. Number two, I want to talk about this here since we are in a theoretical center here. Fisher had theoretical reasons for the importance of small effect genes. And Fisher had a model which is still extremely influential today called this geometric model of adaptation. The idea is this. I have a mutation. It has some effect on fitness. I now tweak it. How likely is it to be adaptive? Well, that's an incredibly complicated problem. If you think about it in terms of, fitness spaces and walking on lattices and all this stuff. But the brilliance of Fisher was he said, we can actually make this into a fairly simple model that captures all the essences without getting bogged down in the biology. And this was his geometric model. I'll show that to you in a second. And Fisher, with this model, showed that mutations of large effect have a low probability of being adaptive, that is favored. Mutations of small effect, as the effect gets smaller and smaller, the chance of them being adaptive approaches 50%. So he suggested, 1930, that mutations of small effect are the stuff of evolution in most cases, which is consistent with his infinitesimal model. So how does that model work? Briefly, it's this. So here, sitting on Z, is the current phenotypic value of my mutation, my allele. Here is some fitness surface. So if I cross this surface towards the middle, I increase fitness. If I go, so this is an isocline of fitness. If I go the other way, I decrease fitness. Here is a mutation effect R. It can occur in some random direction. So Fisher's model is simply, of this arc here, what fraction is inside this circle? So it sounds kind of abstract, but you could actually show that a large number of situations can be approximated by that. And this is often called Fisher's microscope. And his idea is if you're looking at a microscope and it's a little bit fuzzy, if you make a little change in the focus, a little up or a little down, 50-50, it's going to improve the quality of the image. If you crank it way down or crank it way up, it's probably not going to improve that. It's the same idea here, and you can basically show on here, this is for basically 2 dimensions. You can show in n dimensions. So d is the distance between you and the most favorable region. R is the radius, the vector length, you want to do that way of the mutation. You can basically show you get this expression, which would do an error function. So for tiny values, your probability of adaptation is about 1/2. And for large values, there's large effect mutations, you tend to overshoot, and your fixation probability gets smaller and smaller and smaller. If I make this angle really large, the vast majority of that outer circle is on the outside, not inside the circle. That simple geometric argument by Fisher is what's widely been used, and it's one of the justifications for thinking about the infinitesimal model. As we'll see in a little bit, that's not quite correct. So we had all this flurry. Early statistics developed, sort of before the rediscovery of Mendel, but after Darwin. Then we had the rediscovery of Mendel. We had sort of a cantankerous marriage. And then 1918, Fisher kind of came along and sort of resolved a fair bit of that. And that led to things, for example, the classic is Lush's Animal Breeding Plans, published in 1927, which basically founded modern animal breeding using principles of quantitative genetics. Animal breeding has been enormously successful using those techniques. So from the 30s to the 80s, we had kind of a fairly static period in terms of the underlying genetics of quantitative traits. We had lots of development in terms of the theory, in terms of how to use statistical methods to basically improve selection, how to better pick individuals that are going to have more exceptional offspring. At this time, too, of course, this is an extremely active period, right? In this period, we finally realized that DNA was genetic material. We kind of formally proved it with Hershey-Chase. We then kind of figured out the DNA structure. So a lot of molecular biology sort of happened in this period as well. And so you had this divergence between molecular genetics, find the gene, and quantitative genetics, statistically account for the gene. This split was very much like the Biometrician-Mendelian splits of the 1900s, but instead of there being antagonism, it was kind of the molecular geneticists were sort of indifferent. They thought, this is quaint. If you're a pig breeder, maybe it's okay for you, but real modern genetics just doesn't do things this way. But we need a statistician around, so we'll have a quantitative genetics person. So this led to the feeling among many geneticists that quantitative genetics was a bit of anachronism, not suitable for the modern world of today's genetics. Then something happened. What happened was molecular biologists sort of got their act together and figured out how to find lots of markers. And markers led to the kind of, so we had the fields which sort of were diverged in biometricians, Mendelians, came together in Fisher, kind of drifted apart. And then all of a sudden you've got molecular markers here, and then they started coming back together again. So starting in the mid-1980s, there was extensive attempts to map chromosome segments involved in trait variation, so-called quantitative trait loci, or QTL. This idea wasn't new. Payne in 1917 actually did this with long chromosome arms in Drosophila. Sachs in 1923, in a very famous paper, showed that spotted peas tended to be larger, mainly because the allele for spot was associated with the allele for heavy pea size. Today in the 60s did some laborious work moving chromosome segments around Drosophila. But these things are limited by very few markers, very few landmarks on chromosomes. It wasn't until we got lots of genetic markers, and by lots here I mean, wow, I got 50 markers. Now we have, 10 million, we don't blink, but back then 50 markers was a big deal. This then allowed us to do QTL mapping. And I'll quickly go through it here, although most people who's taken the class have seen this exact slide. The idea is if I have linkage, so these things tend to be inherited together unless they break up with fraction C. What I then tend to is if I look at the gametes, that is the egg and sperm produced by this, if you carry a big M, that is the marker, you almost certainly carry one quantitative allele as opposed to the other. So big Q makes you bigger, big M versus little m individuals are larger. You get a marker trait association, and that simple idea is what was used to make crosses and then to track chromosome segments of interest. This was QTL mapping. It was a big, exciting field, still goes on today. Lots of papers were generated by it. Here's one of the first papers, detail papers, Edwards, 1987. And what this shows down here is they looked at a huge number of traits in maize. They had roughly 80 allozyme markers, terrible coverage by today, but it did the job. And they had something like 40 or 50 morphological traits. What this basically plots is the R² value, the fraction of all trait variation accounted for by that chromosome segment. So as you see, these are small, 8%, 4%, whatever. And if you'll notice down here, whoops, that even though there's lower power to detect smaller effects in here, they still had about 60% of all the detected QTLs, accounting for about 1% or less. Now, whether you argue that's more infinitesimal than major gene, I'd say that's more infinitesimal because major genes would be up. So this early paper, which actually was well designed because it had a good sample size, suggested, yes, you could have genes of larger effect, but there are lots of genes of smaller effect. And furthermore, remember, we don't have statistical power down here. There's probably lots of genes up here we can't pick out. The resolution level here was about 0.3%. There could be lots more genes in there, and even though their effects are tiny, if you have enough of them, they could have a dramatic effect overall. You also found genes of major effect. Here's a famous paper where they crossed two lines of monkey flowers that differed in suites for being bee pollinated and being hummingbird pollinated. And what they saw here is, look at these R², right? Big, giant QTLs, big, ginormous R². So things like this, people look at and go, there's major genes in them. Here is kind of a summary from our rather dated summary from our book. So basically, here's the R² value for the largest QTL. So what you see is that, maybe 30% account for 50% or more. That's pretty significant. Here's the R² for all detected. Again, this is down here bizarre because most designs have low power in this area. This is probably much more L-shaped than is suggested here. That just basically says the same thing. So that was the QTL mapping introduction. People got very excited. We can finally take molecular methods and merge them and use to find these mythical underlying sites that involved in quantitative trait variation. Right? That was at least the hope. So from the mid to early 1990s, there was extensive QTL studies, and I call this the era of semi-major genes, because papers like this one and papers finding effects up here, and even things like this where small effects seem to be fairly rare, or less an issue of power, suggest that genes of major effects are common, and they underlie many of the fixed differences between cross lines. So it's important to point out, QTL mapping, I take two lines that are differ in something and I cross them. That's looking at variation in terms of fixed differences. That could fundamentally be different in terms of variation within a single population. So QTLs basically deal with fixed differences. And when they did that, they basically showed genes of major effects are common. They also had a roughly exponential distribution of effects. And you can kind of see that here. If this category is underreported, you basically have kind of an L shape. Again, there's going to be noise because this thing is messy, and differences in sample size because you're putting a lot of experiments together here. But the bottom line is kind of an exponential distribution with a lot of things being small effect and a few things being big effect seem to be the model that really fit the data. Now the question is, that doesn't seem to square with Fisher's geometric model. if I looked at fixed differences between lines, and very often I take a line and a base population, I select it 2 different directions for many, many generations, then I cross them. So a lot of that divergence is going to be selection, presumably. Fisher's model suggests it should be small effects. How can you reconcile this kind of exponential distribution of fixed differences with Fisher's model? Well, Moto Kimura, his name has come up a couple of times. By the way, Tomoka Oda was his student. So Kimura and Oda had numerous papers. The story I heard from Jim Crow was that they had a serious application to put him and Sewell right up for the Nobel Prize in Medicine. And the year they put their application together, Wright died. So they pulled the application. But Wright, Kimura is an enormous figure in population genetics. He noted there was a critical flaw in Fisher's argument. So while mutations with a tiny effect are more likely to be favorable, if that effect is tiny, they're also less likely to be fixed. Those that took the class saw that if an allele has an advantage of S, the chance it's fixed is 2S. So as you decrease, as you make S smaller and smaller and smaller, you're more likely to be adaptive, but you're less likely to be fixed. If you put all that together, you go from Fisher's model here to a model that suggests an intermediate value. That gives you then a more exponential. So Fisher here sort of falls off a little bit quickly. So Kimura is not quite the whole story. It suggests that intermediate effects are important. The one that sort of put it all together was Alan Orr. And Alan Orr basically looked at a model where you're basically doing an adaptive walk. I start here, I try to move to some other fitness peak. How do I do that? And what he showed under very general conditions, using both the Fisher landscape and also models, a lot of this is done using extreme value theory, which is pretty robust. He basically showed that when you walk from some value here towards some optimum, you notice, by the way, the second step is bigger than the first, the distribution of all these segment sizes is basically exponential under pretty general conditions. So the finding that we saw in QTL studies of roughly exponential distribution of effects for things which are fixed, now we can kind of jive it together because we can take the ideas of Fisher and Kimura and Orr and sort of put them all together. That was the happy days of QTL mapping. We kind of understood it. We had some theory. We had big effects. People were happy. Then they started to try to use them for breeding. It didn't work. Then they started to try to map them with big NIH grants. Didn't work. Fine map. What's going on? So poor resolution. A typical design has, you're looking at offspring, you're looking at about 200 to 500 individuals for your QTL mapping. That gives you no closer than 20 centimorgans. 20 centimorgans, roughly speaking, is 20 million base pairs. As I mentioned, basically, it's sort of like saying I've got a gene somewhere in the world, that's the whole genome. QTL mapping tells you it's in India. It's an improvement, it's a big improvement, but it's nowhere near finding the gene. Furthermore, to do this fine mapping, you have to do all sorts of much more laborious crosses like recombinant or nearly isogenic inbred lines. Enormous sample sizes are required. For example, if the QTL map distance is basically 500,000 base pairs, I've got to have a sample size of about 3,500 to detect 10 recombination events. Things to split that up. And that allows me to detect something that has an effect size of about a half a standard deviation. So you need huge sample sizes to fine map genes. The hope initially with QTL mapping is we can use this to find the genes underlying variation, again, move them around and make life really easy for medicine and breeding, etc. Resolution wasn't good enough. Furthermore, much to the chagrin of people that did QTL mapping, and I think I mentioned this before to the class, but for those new people here, so Trudy Mackay was one of the people who have done this, and I think last week she won the Wolf Prize in Agriculture, which is sort of equivalent to the Nobel Prize in Agriculture, for her work on QTL mapping. And one of the things that she found is I have what looks like a chromosome segment that accounts for a big R squared, a major QTL. It fractionates. When I go to fine map it, I put markers in here, split it up. That one big peak becomes two Average size peaks. Okay, 2, no problems. I take an average size peak, I try to map it, it becomes 2 more, smaller. And it kind of fractionates out like that. This is a very common phenomenon we're seeing in mice and Drosophila where you really have the power to do these analyses. So number one, oops, what looked like a big effect turned out to be a bunch of, probably lots of effects in the correlated structure. Number two, there's something called the Beavis effect after Bill Beavis, and I'll show this in a second. Basically, it says that there's a good chance you significantly overestimate the effect of a QTL. And if your power for detecting the effect initially is tiny, that overestimation can be orders of magnitude. So these two together suggest a very misleading view on the nature of quantitative variation, because we thought about major genes because we see big humps and large R squareds. Those both fractionate and also have a good chance of being overestimated. So the QTL view of major genes is now starting to crumble. Again, there are major genes out there, but I'm just saying that as a general view, major genes don't seem to be as exciting as the QTL mapping suggests. Let me quickly explain the Beavis effect. Again, people who took the class have seen this slide. Here is my effect of a particular marker I'm trying to map. There's a variation about any particular realization due to sampling error, et cetera. If I have high power, it means if the values I see is here or above, I declare it to be significant. If I look at the mean of this true value here and the mean of this truncated distribution, they're almost identical. So in high power settings, among those markers I declare to be significant, that is those markers above this line, I can get a pretty close to true estimate of their value. But in low power settings, over here, I've got to be above this threshold to declare it to be significant. Here's the true mean. Most of the time I look at markers like this, they're not significant. When I do, I'm comparing the mean in this tail with the overall mean. That's a pretty big difference. That's the Beavis effect. When power is low, when among those QTLs you declare to be significant, you overestimate the effect. And it's dramatic. Here's Bill's original simulations. We had a bunch of QTLs with each had about a 1.5% effect. Look at the R squared values of those to be declared to be significant. Those look like major genes. Those don't look like major genes. So fractionation, large intervals, and the Beavis effect all led to the realization that QTL mapping, which is this really exciting fusion of molecular biology and quantitative genetics to map the underlying genes, QTL mapping was largely a failure. Largely a failure. You could, I mean, it's very useful in settings. It's useful asking very specific questions, like, are things on the X chromosome? But in terms of trying to find the gene and realizing the genetic basis underlying traits, it really hasn't worked. It also has been very costly. A colleague of mine, Bill Murr, who's a breeder at Purdue, sat down and did a back-of-the-envelope calculation and basically said, for the companies that he consulted for, if they took all that money they spent on mapping QTLs in chickens and simply put that money to having larger raising houses, so you could have slightly stronger selection, that by going to QTL mapping, they've lost over 4 standard deviations of response. It's a big market share. So QTL mapping was kind of I would call it an epic fail of quantitative genetics. Well-intentioned, really misled the field for a while. Another issue, let me skip that. I'll come back to that. That's interesting. I'll come back. So it looks kind of bleak. But the molecular biologists kept on toiling, and they went from now getting allozymes to now getting SSRs. All of a sudden, they could get SNPs. They could get pretty easily 100,000 markers, 1 million markers. And this opened up an ability to do a different type of analysis called association mapping. QTL mapping, I've got across 2 populations. So QTL mapping is basically picking up the distribution size of fixed differences. Association mapping is going in and sampling from segregating variation or population and trying to find the effects. Let me kind of walk you through that again. So the idea is to look at marker trait associations. I have a marker. I look at individuals with a certain configuration of that marker, look at their mean, look at individuals that same marker with another configuration. That is, one's a homozygote, one's a heterozygote. I ask, is there a statistically significant difference between individuals with different alleles of that marker? If there is, That's a marker trait association. That marker is linked to a gene underlying the trait of interest. Difference here is we're looking at a population sample. These can be much larger. Nowadays, a modern association study for human genetics has 100,000 cases, 100,000 controls, and looks at 1 to 5 million SNPs in every single individual. That's a big-ass experiment. When you do that, the level of resolution depends upon what's called the linkage disequilibrium. If I look at a chromosome region, are there non-random associations? It turns out there are. In humans, these are on the order between your population of 1 to 10 kilobase, that is 1 to 10 kb is 1,000 bases. They're under 1,000 bases if you look in maize, and Wolfgang and Drosophila LD is what, about 500 or 100 nucleotides? Is tiny. Yeah, so level of resolution here is much, much finer, going from a megabase scale to a kilobase scale, and sometimes hundreds of base scales. So number one, we've got segregating, we're sampling variation in an existing population. Number two, we've got a much finer level of resolution. How does it work? Really simple. This circle is a population. The red lines are different types of chromosomes. Now a mutation appears on this red background. Whenever I see the mutation, I'll see a red background, unless I've got recombination. With 100,000 markers, markers are incredibly close to any given random site on the genome, so it's going to take a really long time for that to be scrambled up. That creates what's called linkage disequilibrium, a non-random association, and that signal will persist for thousands of generations. And that's what we use. And we can tell how important that's going to be by simply looking at our organism, picking random markers, and figuring out what LD is. In humans, it's on the order of a kilobase. In wild maize, it's under 1,000 bases. In elite lines of maize, those blocks are 100 kb long. So I can look at an organism, sample markers, and tell me what the level of resolution is going to be before I do the experiment. Here's kind of how it works. Each dot is a marker that I test. I'm testing 1 million markers, that's why it looks all fixed in here. Everything above a threshold is then declared to be significant, and that now is a region, instead of being megabases, is a region that's maybe a kilobase. So now I'm either within a gene, so I've got a 4 or 5 kilobase gene. I've now got not just within a gene, within a small region of that gene. So I'm not at the nucleotide, but I'm a lot closer level of resolution than I was before. So it's much more powerful, much more fine scale, easy to do, and now we have lots of markers. It's easy to carry out. So people did that. And when they did that, they often did the following. Okay, how good does it work? So they take something like height. They can do an association mapping for height. We know from years and years and years of studies on twins that if you look at the height between identical twins, and that is monozygotic, and non-identical, that is dizygotic twins, we can estimate the fraction of all variation that's genetic. And in twins, it's about 90% if you control for sex. 90% additive, which basically means single alleles at a time. Very, very high. You then go and take all the significant SNPs you have, estimate their effects, put them into a regression, and you can account for 10% of all the variation instead of 90%. The consternation was, where's this missing heritability? Basically, this is sort of all the guilty people in the field. I'm just kidding about that. So this paper here in Nature, this isn't what made the topic interesting. What made the topic interesting is this, the picture. The caption was, the case of the missing heritability, with little burglars sneaking out the back window with the heritability and the shocked investigators. So this led to a large number of papers. Again, wasted efforts, I'll talk about in a second. And then the molecular biologists said, well, obviously this again is a vestige of the Mendelians. Well, we know about genetics. We don't want these statistical descriptions because we know they're flawed. We're not getting at the essence. We can get at the essence. And therefore, there's something wrong with quantitative genetics because we have the genes, and yet these methods of looking at, SIBs, twins, those are clearly way off. So obviously the problem was with those methods, not with us. We have all this NIH money that we get, billions of dollars to map it, so obviously we're in the right. What's behind this missing heritability? Let me show you what's behind the missing heritability. It's the fact that they didn't take a quantitative genetics course. So let's imagine I've got a polymorphic site. Big Q and little q are two alleles that influence the trait. Again, if you've taken the class, you've seen this slide before. So let's imagine now, here it is, I have a mutation that takes little q to big Q, and it arises on a chromosome right nearby it. I've got the big M allele. So big M has frequency P in the population. The big Q allele has effect A, little q allele has effect 0. Among chromosomes that carry big M, R of them carry big Q, the rest carry little q. So I've got this association. And this is as strong as I can make one for detecting this. When I go ahead and do that, and I basically ask, what's the effect, I can compute how much variation in the trait is caused by the causal site. If I knew Q exactly, I'd get an expression for how much variation I see. If instead, I look at how much variation I see is accounted for by the linked site, the marker variation, I get an effect here. Their ratio is R. R basically is the fraction of M that carry big Q. If this is tiny, even if you pick out every single significant marker, you're only going to account for a tiny fraction of all the variation. So if alleles tend to be fairly rare, you're not going to pick them up. Number two, let's go back over here. Number two, these are the only markers I put in my analysis because they're above the threshold. All these markers down here are probably biologically important, but because you're making millions of tests, there's such a high stringency, you throw them out. Those two things together, multiple tests and the fact you only account for a small fraction, take care of the missing heritability problem. Low power, yes. Hang on, this one here, yeah. Oh, those little arrows are genes that the researchers found endearing because their lab works on them. Those are basically, these are calls for SNPs they think are important. And I'm not sure why they're ignoring these others, but these have got some story why they're important. This is a typical GWAS. It's called the Manhattan plot. You see things like this a lot. Each is a significant marker, and it's put into a regression. When you put all the markers in a mixed model framework, you account for about 40% of the variation. And that's, remember, and then each marker then would also only account for some fraction R of the actual variance. So missing heritability is not an issue at all. People got all excited because they didn't understand basic quantitative genetics. Again, this riff sort of comes back to bite us a little. So we now have lots of GWAS studies which are done. What are the take-home messages from them? Number one, many sites, each of which has a small effect. So it's almost infinitesimal-like, but it's a bit different here. Because when I talk about a site having a small effect, I go to a SNP, it means the variance that SNP accounts for is tiny. Well, that variance it accounts for, let me go back over here, is this thing right here. That's the variance. So if the allele is rare, that is, this frequency is really tiny, that effect could be really large, but I still have a small variance. The view we kind of have now is it's not an infinitesimal effects, it's an infinitesimal variance, that each site has a small variance. It doesn't mean that alleles there are tiny, it just means that if they're big, they're rare. And as we'll see in a second, that's often what you see. Why? A big allele often affects many traits, and therefore it's selected against, so its frequency is going to be low. So the GWAS suggests that you've got lots of sites, each of which accounts for a small fraction of variation. But again, small variance doesn't mean you don't have large effects. Number two, hits in non-coding regions are at least as important, at least as frequent as hits in coding regions. So not surprisingly, regulatory variation is important. In fact, the general view is that regulatory variation is probably the most important thing. Why? Much larger mutational target size, and also much more fine-tuning. You change an amino acid, things can get pretty wonky. You basically change a single base, so the binding goes up or down a little bit, you can fine-tune it a bit. So the GWAS studies suggest infinitesimal-like in terms of variance. Regulatory variation is important. But here's an example from height theta. What you see for height is that if you have a small effect, they tend to be pretty frequent. If you have a big effect, log scale here, they tend to be pretty rare. This kind of ghost region in the middle is the design, because if you've got a rare effect, you can detect it if it's big enough, if it's rare. If you've got a small effect, you can detect it if it's common enough. If it's kind of intermediate, the sample sizes aren't big enough to detect this. So a lot of the things we think are small effect sites are small effect in the variance, and they could be big alleles sitting down there at low frequency. So again, the variance accounted for these are small, but we'll see in a second that doesn't mean that they're ecologically or evolutionarily trivial, because that's potential genes to source out when you undergo selection, as I'll show in a second. So what's the current view of the sort of Fisher's infinitesimal model? So most detected variants have only a small effects. A lot of really excellent work has been done in maize. In fact, some of the best work has been done by Ed Buckler's lab in maize. So the same loci are often used over lines, but with different alleles. So if I'm looking for a QTL for water stress, if I look across different lines, I find alleles for it. And those alleles tend, not always, but tend to be in the same genes, but you have novel alleles in each environment. So a new mutation comes up, it's probably novel alleles, and oftentimes it's in the same site. So because of this fact that if I look across a chromosome, I've got a whole bunch of small regions of all roughly equal effect, it now means that if I'm trying to do genetics for disease or genetics for breeding, what doesn't matter is individual genes. What matters is how much of my total genome do you share? Because under the view that you kind of have equal effects all the way around, it doesn't matter which segment you share. If we share 50%, it doesn't matter which 50% we share. If there's no major genes, those are all roughly equal. And what this means is the fraction of shared identity is all that's needed. And what this basically points out here is we can estimate that with more precision using molecular data. The old way was estimating it by expected value for a pedigree. If you and I are full SIBs, we should share half our genes, but there's some variation about that. If I actually use molecular data, I can actually calculate that directly. And this is the method that's used by things like genomic selection, which is now the new method that's kind of replaced QTL mapping. And in essence, what you do is you scan two individuals' genomes, you look at what fraction of the genomes that they share, and then that basically tells you how similar they should be. That effect is also then used, by the way, in what's called molecular medicine, where I'm trying to, instead of predict an offspring, predict your phenotype given your genotype. So what's the current view of quantitative genetic variation, to summarize here? Number one, most traits show significant additive variation. That means individual alleles by themselves contribute a lot, not their combinations. Number two, there are many genes of small effects, in quotes, underlying the variation. These are either small effect alleles at modest frequency or major alleles at low frequency. There could be an exponential distribution of effect size, but it's masked by this inverse relationship between effect size and frequency. So what I'm plotting here on the GWAS is variance. If I plot allelic effects, I might see something quite different. But rare alleles tend to be large. Common alleles tend to be small. So I'm kind of masking that. Regulatory regions are at least as important as structural regions. Much of the low frequency variation is likely due to novel mutations. So suppose I'm looking for genes for asthma. You can look at target candidate genes in asthma, and what you can see is that those genes are enriched for rare mutations. I take a pool of individuals with asthma, a pool without asthma. The pool with asthma has lots more rare mutants, but they're not shared. So any given mutant isn't predictive, but we do know that basically if you have asthma, there's a better than average chance you have more mutations in one region. So that's not good enough in terms of predicting what's going to cause variation, but it suggests that a lot of variation is caused by novel mutations. That's exciting, but it's also disconcerting. It means you don't have a lot of predictive power. I didn't talk about this, but basically, if you do the extremely careful studies in Drosophila, where you basically change out a single nucleotide you've nailed down, or a single chromosome region, really, really tiny, single gene size or less, and look at those, you often see epistasis, nonlinear interactions. But if you look at that's because you do them in an identical inbred line background. You try one combination, another combination, you see epistasis pretty strongly, genetic epistasis. But if you put those combinations in random backgrounds, you don't see epistasis. And what that means is that in any particular genetic background, you can see epistasis, but because all the genetic backgrounds are randomized, no two individuals have the same background, that epistasis that's there gets washed out, you're simply left with an average effect. Put it another way, epistasis is so widespread you can ignore it, because each genotype is unique, and therefore you can never predict it, and therefore you simply look at the average effects you're looking at. And that's kind of one of the take-home messages. So the marginal average effects, its additive value, are what's important, is most of these epistatic effects tend to be small. So what are the implications then of this view for a couple of things to finish off? Number one, tests of selection, right? Wolfgang in class has been talking about these. It's a big pursuit. So if you have adaptation from standing variation, as opposed to novel variation, you get a much weaker or no signal. Number two, if you have small effect genes, they have a small selection coefficient, you get a weak signal. But number three, if you reach in and pick out some of these really rare alleles in the tail that are of big effect, those could have a significant signature in terms of selective sweep. So the view of how that variation is distributed impacts that. That just basically shows an example of the sweep. This gets at your question. Selection response. If major alleles of large effect are rare, if my population is big enough, I can exploit them. Most breeding populations are too small. And breeding populations are set by the number of adults you use to make them, not their actual size. So here's an example of that. Here is, if I basically run through and get a distribution of allelic effects like I see there, and then I select, here is what the heritability increases a little bit and then goes back down. So in this example here, that wouldn't bother you at all. But look at this example here. Here I have allelic effects in their frequency. Those are unlikely to be grabbed in a selection experiment because they're really tiny, or in a lot of domesticated experiments. If I use that full distribution, look what the heritability does. Why? This is a rare allele increasing in frequency, which increases the heritability. If you saw that with these bursts of response, you'd probably say it's epistasis or it's new mutation. And what it really is really rare alleles at low frequency. As they increase, they inflate the variance. Then they start decreasing the variance. So if you have a really big population and alleles of big effects are rare, you expect to see this very uneven. Like, look at this after 80 generations. That's not a new mutation. That's a really rare allele of a really big effect that finally is at enough frequency that it has a big impact. So this new view of quantitative variation changes how we think about selection response. In particular, you want to keep the population size as large as possible. Why? All these big effect mutations down there will be lost by sampling in a small population. If I grab 100 individuals from this population, I'm unlikely to grab most of these. If I've got a really big population, there's a good chance I grab those, and those could be really important for breeding later on. What about disease risk? Same thing here. If the goal is to talk about personalized medicine, it's a bit disappointing, because you can imagine a lot of disease variation is caused by new alleles that are either restricted to a specific pedigree, there's a family group, and not shared over the population. So you won't be able to then look for markers in those individuals. All you can do is basically, if someone comes in and asks, what's my cancer risk? You can scan them, and you can say, oh, you've got mutations in around a region we think may be involved in cancer. If the mutation knocks out a gene, we can tell that. That's pretty easy if it knocks out a coding region. But if it's just a SNP in there, there's lots of SNPs, lots of variation. So this idea that a lot of mutations are novel that cause variation means that the view the Genome Project was sold on, personalized medicine, I can read your genome. I can tell you what day you're going to die, who you're going to marry, and how much you're going to buy, and what your car is going to be in 10 years, right? If a lot of that's novel variation, we can't pick out. So finally, what's the present role of quantitative genetics? Well, in the genomics era, it's as important as ever. Why? Because it basically manages all the uncertainty. We have explicit understanding of some simple traits, but our knowledge of complex traits is lagging. we can put this information from simple traits in, and quantitative genetics will basically count for all the uncertainty in the model. That's sort of what it does. Basically, last slide here, the framework that does this is kind of the workhorse now, which is called the general mixed model. And this is the framework that underlies most analysis in quantitative genetics today, things like association mapping, things like genomic selection. And basically, this is where we incorporate genes of known effects. This is where we incorporate background things we only know variances about. So that is your current view from day one up to kind of state-of-the-art in quantitative genetics. And I have like 48 seconds for questions. So, questions, comments, disputes?
You think those are just outliers?
Yes, so they're probably outliers. So certainly things like, there are super genes which are locked in, like butterfly mimetic chromosome patterns. So whenever I say these are kind of generalizations, right? There's no clear, what this means is if I pick a random combination, I might likely see epistasis. Well, so epistasis is kind of like, so it's kind of like, remember you took a calculus class and you didn't quite know what the answer is, but you figured it had to involve pi, so you put pi in there and hoped you guessed it was a quarter or a sixth or an eighth. That's what epistasis is, people don't understand. It's like, well, things are really complicated, it must be epistasis. And in fact, it turns out you can account for a lot of these complicated things without epistasis. Epistasis is hard to measure, it's not involved in breeding, because it just gets reshuffled each generation. So molecular biologists love epistasis because it's kind of, if you don't understand something, you say epistasis. But it's actually not very practical. Wolfgang.
So Trudy McKay, she thinks that the SNPs have a lot of genetic epistasis. Yeah? So you say we shouldn't even worry about it because it all is in the wash.
Well, so I went through the argument fairly quickly. So what Trudy has found is, she's found if you do the following experiment, you get a background where everybody's genetically identical, and then you put in 2 genes that you know are involved in the trait. Mm-hmm. And you put in different combinations of those 2 genes in an otherwise identical background. Mm-hmm. In that case, you see non-additive interaction between many combinations, epistasis. Mm-hmm. However, if you do that same experiment and don't use an isogenic background, where everybody's the same, but put them into a random mating population, there's no signal. What that suggests to me is that there is epistasis. You see it in that background, but that's one particular background. As you change background, you change that epistatic pattern. Every background in an outbred population is unique. Therefore, there's lots of epistasis, but none of it carries over across random. Most of it doesn't carry over when you randomize over genomes. In genome A, you see it. Genome B, you see a different pattern. Genome C, a different pattern. And that averages out because it's the entire background that matters. And that's the entire genome.
So is this the explanation that in one data set, she finds this panel of SNPs, and the other data set she finds this?
Yep, so that's the epistasis. But those are done in isogenic backgrounds.
There's no overlap.
There's often very little overlap. But the fact is, the point that makes it for me is that you do this experiment so all of you have the same background. I do this experiment, I get a big signal. Now if I do that same experiment, but now you have random genomes, there's no signal. What that suggests is there's obviously a clear epistasis in that background. Nothing special about that background. It just suggests that it's very background dependent. Each of our backgrounds are different. All that, for most genes, kind of averages out. And I simply pick up average effects there. So the way I phrase that, to be provocative, is epistasis is so unimportant because it's so widespread.
Why did she get the prize?
Well, because she's done lots of good work. Why, Wolfgang, did you finish second in your bidder? Is that what you're saying?
Can you elaborate a bit more on what you said about epistasis is so common that it's unimportant?
Sure. It's a provocative statement, absolutely. And of course, you know me, I want to be a bridge maker and not. So no. So I'm actually giving the opening talk at the World Congress in Quantitative Genetics, and the title of that talk is called "Epic Fails in Quantitative Genetics." So I'm certainly going to be provocative there. So the idea is this. If you take gene A and gene B, so if you look at big A, big A, big B in one background, you see strong epistasis. So I've got an isogenic background. I put big A, big B in there. Big A, I look at lifespan. I put big B, I look at lifespan. I put big A and big B in there, I see a different, there's not the sum of the two. I see a strongly epistatic interaction in an isogenic background. If I repeat that same experiment, not in an isogenic background, but over a random background, so everybody here gets a big A, I look at lifespan. Everybody here gets a big B, I look at lifespan. Everybody here gets a big A, big B, I don't see interactions. And that's because in the isogenic experiment, everybody has the same background. In the other experiment, all the backgrounds meaning the rest of the genome is different. So you see epistasis when you fix the genome, the whole genome. These two in a particular combination of genomes show an epistatic pattern. If you randomize those over all genomes, which is what random mating does, that epistasis goes away. So because you're averaging it out.
So I understand that.
Okay.
But then why limit the epistasis argument to just two loci? One could extend that to just saying that basically whatever else is in the background that you're randomizing,
Right.
there's some bits and pieces there which maybe have like a 3 or 4 or whatever.
And no two individuals in an outbred population will have the same background, therefore you can't propagate that epistatic signal. So it's not just the two players involved, it's everybody they interact with.
Sure.
In a random mating population, all that washes out because it gets randomized. So the net
So doesn't that actually prove the point that every individual
exactly,
is going to
epistasis is so
point.
widespread that it's unimportant.
Ah.
That's the point.
Okay.
I'm not saying it doesn't exist. I'm saying it's so widespread. If, for example, here's epistasis. If you always saw it independent of your background, you'd see an effect. But it's so background dependent, everybody's got a different background.
So maybe
Epistasis goes away. I mean, it largely goes away because it cancels out.
basically you have to redefine epistasis as between various different genes and loci then instead of just 2 or 3. No, but I'm trying to think of like maybe it's not that it's not important, it's that it's important but it's hard to predict.
Well, but here's the point though. The point is that
Useful. That I agree with.
see, epistasis puts us in the dark here. So good time to answer this question because we're in the dark about it. So people get hung up on epistasis. The question is it useful for predicting evolutionary response? No. Where it is useful is asexual systems where you can propagate. So for example, in Isabel's virus work, epistasis can be very important, right? Because you can propagate those genomes. But in sexual species where you're scrambling your genomes all the time, what you have to ask is, do these two combinations have a predictable interactive signal when I randomize them over genomes? Turns out that they don't. So that's how you would see epistasis if you were evolution or a breeder or evolutionary biology. If you're a molecular biologist and are fixing all the components, you see there's an interaction, and that tells you something about it. And it probably comes down to these systems biology arguments about robustness, but if you fix all the elements in network, which is what the fixed genotype does, you may see one pattern, but if you take that network and randomize it over all combinations, that pattern may sort of average out and go away.
Yeah, but again, when a population is just, sorry, if anybody else has any questions, just
That's okay. This is
we can do something else.
I mean, Deepa, as you know, not very stubborn. She'll just back away from this.
No.
Okay.
I'll talk to you later. But
That's called sarcasm,
I
by
have
the
more
way.
questions. I don't want to hawk the whole time.
Yeah, so that's a good point to discuss. Again, it's provocative, but doesn't mean it's wrong. It's not salsa dancing, but you know. Other questions or comments? So Wolfgang said all the models should forget about epistasis. I didn't say that. I said that I think that people get overly hung up on epistasis. So it comes down to this. What's the question you're asking? If you're trying to exploit genetic variation for breeding in an outbred population, epistasis doesn't matter because you can't propagate it. It gets randomized each generation. If you could estimate it, which is difficult because combinations are in a small fraction, make it very hard to estimate, then if it really context dependent, you have to re-estimate it every generation. You're much better off just spending that money and emphasis on larger populations, you get a larger response. In terms of predicting for medicine, if each epistatic combination is unique, I can't look at your genome and tell what your epistasis between two genes is going to be, because each combination is unique and we have all these novel mutations. Each of us differ from each other by about 22 million nucleotides. And we share some of those, but View and I are about 22 million. So in a small sample, there'll be a lot of things we differ by. Very hard to pick those up. So epistasis certainly may have an important, because it might be that in you, you've got the right epistatic combinations with all the background. But that's unique to you, and no other person in the history of the planet would ever have that. So why is it important to know about that? Because it has no predictive value. I can never do science on it unless I want to replicate you. That's a scary thought. Have a bunch of little Wolfgangs walking around. But so that's why I think epistasis, you can ignore it because it's so widespread. Now, if your question is, I'm trying to understand a particular pathway, and I've got isogenic lines that I can do that with, then epistasis may actually give you lots of insight. Because you look at different combinations and see what they tweak and what they involve. But when you infer a network, you infer a network by perturbing it and look at a combination over random individuals. If you took a network and perturbed it environmentally, that would only give you a small section of what it's going to do. So take an inbred line like yeast, perturb the network from an environment, that tells you something, but you also want to see how that network is perturbed when you bring in new mutations. So I think epistasis is incredibly widespread. I also think for many problems it's really not that important because it appears to average out. Doesn't, I mean, it could arise in certain situations, and probably does, and there are cases where you see lots of epistasis, but people get so hung up on epistasis as opposed to things like additive values of traits, which are far more important, and far more explanatory. It's not as sexy as epistasis.
Low frequency. I have the large effect allele sitting in sort of the low frequency range, and so how can I get them to sweep?
Great question. How can you rescue those low frequency alleles? Your friend is now, if they are, so they're typically there because they have pleiotropic effects. Let's say you're looking at height. You've got alleles there that increase height, but they do a bunch of stuff. So that low frequency getting knocked out and kicked back in by mutation selection balance. If now I select for height, and select strongly for height, I can, with a big enough population size, A, they'll be in that sample, and B, 4NSP will be significant enough I can reach down and pick them up and start to move them. So the answer here is quite simple. Large populations.
Shifting the optimum.
No, well, no, it's basically, if you want to exploit them for breeding, just make sure you have a large population. If they're evolution, right, when your peak shifts, those now can come on in like you saw in your curve, right? Microphone over here. Let's pass the mic. Wolfgang's got two, one for each hand.
Is there a general number to that large population? I mean, can you say like an estimate of how large is large?
You can do that.
Space?
So basically, if you remember, we put Kimura's diffusion equation for fixation probability. The thing on the top was 1 minus e to the minus 4NESP. P we take as 1 over 2n, get 2s on the top. So the thing to look at is 4NESP, meaning your frequency. When that's large, selection dominates. So that's actually less stringent than 2s because it accounts the frequency. So that what basically sets it. So what you can ask is, suppose I want to have, remember what goes into that equation? It's S times P. So you can look at what NE do I need to get an SP product sufficiently large. So if SP is, 10 to the minus 3, you need a population size of about 1,000. That's effective population size, not breeding size, or not census size.
In the Manhattan plot that you showed, you said that most of the polymorphisms which affect the variance are below the p-value cutoff. So are you essentially arguing in favor of reducing the stringency of these studies?
So that's a great question. So how do you handle data sets like that? So when you do test them one at a time, you run these huge problems. If, however, you do the following, it's a random model. Each SNP has some value drawn from a distribution. Okay? Then I can put all the SNPs in my model. I can then estimate the variance of that distribution, And then I can predict each SNP value. When you do that, doing all the SNPs at once simultaneously, that model then accounts for 40% of the variation. So I'm not caring about the SNP effects. I'm just putting them all in and saying how much does it account for. You can then use that model then to show what the actual SNP effects are. It doesn't give you a p-value, but it says if I put them all in, how much do I account for? And again, you have to put them in a random effects framework. You can't put them in as fixed effects for another value. But random effects are cool because how do people analyze a microarray with 30,000 genes and 10 replicates? You don't have 30,000 degrees of freedom, you have 10. But in a random model setting, in a random model, what I'm doing is I'm drawing things from some distribution. So every observation, every gene gives me information about that distribution. What do I estimate? I estimate one thing, its variance. So 1 degree of freedom in a random effects model lets me predict 30,000 or 100,000 or 600,000 or 4 million things, assuming the model is good. And you can fiddle with it and stuff like that. So that's how people do those. Folks that have done those are folks in University of Queensland, like Peter Vischer and Mike Goddard and Ben Hayes. And by the way, that's the flip side of something you might have heard about called genomic selection. Association mapping and genomic selection are equivalent. Just looking at the mixed models differently. Other questions? For the class, my lecture 10, I'll put, hopefully tomorrow morning, I'll have the lecture notes sent to the webmaster. Right now there, what I've posted is the background information, more than you ever need. That lecture is on neutral, if I see traits diverge, how do I test for neutrality? There's a big chapter in the book on that, which is more complicated than you need, but it's there for background if you want to go into some more of these stuff after I get the lecture. So it's there, feel free to download it.